Hello, everyone. Good morning. It's 8:30, so 8:31, so we're going to get this party started here. Welcome to the symposium number 17. This is the Arctos Symposium. There's also a bunch of other database ones, but I'm happy to see all of you guys here. I just want to make sure you're in the right room. So you're in the best room, exactly. So good morning and welcome. Thank you for coming to the Spnhc conference. I hope everyone recovered from the heavy dancing from last night. That was quite a blast from the past. I also wanted to make sure we thank and you guys all had a chance to visit the sponsors upstairs. And now we're going to get started. So as a way of intro, just like all of our other Arctos community events, so majority of you guys are current users. Hopefully we'll have a few new users, but we always like to make sure this is an open conversation. So we're going to have a series of presentations. Let me see if I can get my thing started here. Whoops. Sorry, we're not used to PCs here. And we're going to make sure that people get a chance to also ask plenty of questions at the end during our panel discussion. Why are we not getting into slideshow mode? Oh, it is in slideshow. Oh, I apologize. Okay, so obviously, yeah, you guys are seeing. So, like I said, this is a quick overview in my introductory talk. There we go. No, advance. Yeah, that's what I'm trying to do. It's not letting me do that. I probably unpresented myself. How do I advance? Okay. Oh, so let him.
Yeah, if you bump this cable, it doesn't like it sometimes.
Okay. It's the J-Hawk mystery room. Oh, now, wow, we've really.
So which PowerPoint will come up now?
Well, we can start in the middle too. That's fine. It's all one big giant network universe. Okay, so now I can use advanced. Okay. Thank you. So, like I said, welcome to the Arctos-verse. You can see here a list of speakers today. My name is Michelle Koo. I'm the director for the Arctos Consortium. I'm also a curator at the Museum of Vertebrate Zoology at UC Berkeley. I'm going to introduce each of our speakers as we go along, but the names with the stars are part of the executive committee of Arctos, the officers we like to call ourselves. And I also want to thank right up front to the entire Arctos working group, especially the members who engage us on our public forums and in our monthly meetings, as well as our technical team, Dusty McDonald, Joyce Gross, Deanna Lovett, Erica Krimmel, and Teresa Mayfield-Meyer. And so you can see my quick overview, so this is going to go a bit quick, hopefully. Oops, there we go. So who is Arctos? So we are a consortium of member institutions. This is just some quick graphics so you can see how we are distributed around the world, mostly in North America, but we're now in 8 countries. We represent about 3 main collection types: biological, geological, and cultural. You can see by majority of us are biological collections. We're also a community of practice. We happen to share a database, and so we meet regularly each month in various online forums, so we can share ideas, best practices, bring up maybe unusual collection issues, but also discuss and improve the database. And it's also a great place to learn about other collection types and think about ourselves as museum professionals. The basic Arctos governance model is one of community. So this is just a quick graphic interpretation of how we have a central day-to-day operational executive committee made up of the officers who interact directly with the Arctos members and users, and we're overseen by a board of directors that we call the Arctos Advisory Committee. Arctos loves diversity. I'm going to be able to say the word diversity, I think, at least for now. We have records of all types. And again, this is a fairly good representation of the kind of collection types we have, but also the record types that we have. So many of our consortium members actually have more than one kind of collection type. So they may have majority biological, but also some cultural and maybe a zoological collection as well. And they're all kind of growing in number, but this is a pretty good, this is the split by record type. You can see we have all sorts of kinds of records and different kinds of bases of records, and each one of those records can be linked between each other, and we're going to talk a lot about those linkages. The other kind of diversity we represent are these sort of topical thematic interests, and so many of us are also part of committees and subcommittees, sometimes ad hoc committees, that tackle on specific topics, disciplines, and those are brought back oftentimes to the larger Arctos working group for discussion. So I'm going to spend a little bit of time talking about our development approach and sort of our philosophy around these things. And so in general, this is like the highest level of what guides our approach to the database. So we prioritize the semantic web. We've been doing this from our inception. So that means, what that means is we love linked open data, URLs, URIs. That's how we enable the extended specimen concept. But like I said, it just makes sense because the internet is full of more resources than one database could ever have. So we like to link all of those so every record has as much information in one spot for the users and for the curators. We also prioritize data normalization. So Arctos relies heavily on code tables. All of those code table values need to be have accepted at least definitions by the community or rely on some kind of external authority. So we look for those. And that model has served us well because that's been allowed us to actually start accommodating all these new kinds of collection types. And we really haven't encountered a kind of collection that we could not accommodate so far. So with all this in place, the other last aspect is relationships. And so those are those linkages, mostly internal, but also external to other, maybe a third-party repository. So you'll see more examples of that. This is just sort of some big bullet points about how we operate our cyber infrastructure approach. So we have a strong partner at the Texas Advanced Computing Center at TAC. We are in a shared environment, and that means for us that there's a lot of shared benefits. That means we have a single pipeline to aggregators, thanks to the VertNet IPT. We also then have sort of shared pipelines to other third-party and related resources, not just GBIF and GGBN, but also like iDigBio, IsoBank, and a number of other ones. And you'll see some examples of that today. All of this ends up promoting data quality, all of our development principles. But the main priorities are all community driven. So that's why we need to have those monthly meetings and regular communication with everyone. All of this is managed at a collection level permissions. So that means that we can have multiple roles, but they're all defined by which collection allows views and management to specific users. I'm also going to take about like 3 slides to give everyone the loved for everything. I think that what a lot of people were here for was the NSF grant update. So yes, we are still in our no cost extension, but this is our last year. So this will be the last NSF SABI update. But for those who are maybe unfamiliar with this grant, it gave us 2 things to make us sustainable. One, the resources to focus on our infrastructure, so I'm going to talk about how we met those goals, and then the sort of financial administrative end of making Arctos sustainable. So on the technical side, in the first year, the major lift was migrating from Oracle to Postgres. So that came with, a lot of testing, a lot of exploring options, but eventually we settled on this, and this was like a major effort on our technical team and I should say the users. So I appreciate everyone's patience with that process. In the second year, we started focusing on the core functionality and expanding our collection types. And then we started working with specific collections and migrating specific functionalities. And obviously, this is an ongoing process, so we're still working on accessibility and a number of requirements moving forward into the futures. But we really feel good about our decisions and approach to the larger cyber infrastructure for Arctos. On the second deliverable, and that's more the administrative organizational sustainability. And we learned a lot about our NSF too, I should say, about what they can and cannot fund. So in our first year, what we did was form a 8-member task force that was composed of a few officers, some board members, and some users. And we explored all different options and business models. What we ended up settling on was a fiscal sponsorship by the Community Initiatives, which is a nonprofit based in Oakland, California. And they focus primarily on being fiscal sponsors to enable other projects to move forward and not have to worry about, the administrative end of running a nonprofit. So we can have all the benefits without all the burden. So that's worked out really well, but it also meant for us a bit of a learning curve, how to hire through this new system, how to transition staff that were 100% on a grant at one point and now are 100% on Arctos's payroll. So we're continuing to grow in our membership, even though we're not necessarily actively, recruiting, for instance. So it's still been an exciting journey, and I think we're really set up well to continue with our growth in members and collection types. So here's a quick snapshot of some of the members that we brought on during this NSF period. It's not all of them. We had, I couldn't fit 42 logos on here, but, and if you guys got a chance to see the Portuguese Blue Biobank poster upstairs, you were introduced to one of our newest members. So what's going on for the future? Everybody wants to see this slide. So this is just a quick snapshot of some of the things that we're working on right now. We're experimenting with different kinds of access, how to make Arctos more accessible. We had a UC Davis Masters of Business Administration team work on both an assistive chatbot as well as some new dashboard interfaces. The chatbot's going to take natural language processing to convert a query like, where are all the frogs in Kansas in your collection into search parameters to make that a little bit easier for people who maybe are not technical or have some limitations using the interface. We're also looking at Elasticsearch as a new kind of filterable portal. There is already an Arctos-R project that was actually built by a student here under the mentorship of Marlon Kobos and Vijay Barva, which I don't know if he's here today, but so we're going to continue working on the Arctos-R project for researchers. And like I said, we're also continuing to explore some data visualization widgets. And so you'll be seeing some new changes coming up to the interface soon. So this symposium, like I said, I'll just give you a quick intro about what you can expect. You're going to see Arctos in action. You're going to see Arctos in use for a number of different kind of collections. So we're going to have a fly-through demo from Emily. We're going to learn about object tracking from Andrew Dahl over there. We're going to hear about relationships from Mariel Campbell remotely, and then see some demos from Vicky Zhuang. We're going to hear from the Oh, the whole OGL crowd over here. I think, Hannah, you're giving the talk, so we'll be hearing about the marine genomic resources, and we'll also be talking about how you can, how Arctos tracks impact. And we'd like to use up as much of the presenters' time in presentation, but we're going to have an extended period for panel discussions. So be prepared for that. Save all your questions and good ideas so we can hear from you guys. So onward. Let's get into the Arctos-verse. Thank you very much. Oh, yeah. Okay, sure. Thanks. I don't know how to get into the next one. That'd be good. They're all loaded up at the bottom.
Okay.
The PowerPoints. Okay.
Yeah. All right. Thanks, everyone. So we are going to take a very rapid journey through Arctos, so buckle up. I'm first going to demonstrate some of the public-facing user portal before I log in, and then you can see some of the operator permissions. So we are on the main search page here. This is really akin to a simple search, but you can see I have access to the 5.5 million records in Arctos. But I wanted to start by saying that this form is highly customizable. So if I go to this menu here, we see there are a lot of presets available that really fall along discipline lines. So for instance, if I click Advanced, you can see all these categories really build out, and this lends a lot of precision to searching. So for instance, if I look at locality attributes, you'll see there's a lot of structured data around paleocontext, as well as if I go to record attributes, a lot of controlled vocabulary, mainly around trait data. And I'll also add that this form is totally customizable, so you can actually toggle on and off exactly what fields you want displayed, as well as how you want your results to be structured. So, and that you can save to your profile and get exactly what you want. So I have queued up a search so I don't have to type in front of you all. But we're going to search for deer mice that have been examined for hantavirus. So this is controlled vocabulary. And again, we're searching across Arctos because I'm not logged in and limited to my records. So we'll fetch this data. And maybe I'll just do an interpretive dance while we're waiting for this to return. But yeah, so here we've got results. We've got about 1,500 records, and obviously if I click on that link, that will navigate me to those records. But I did want to show you a nice tool where you can actually summarize how your results are displayed. So if I'm interested in filtering by state and county, now I have my 1,500 results available in 30 rows, so then I can open that faceted search. And those parameters are customizable as well, so just a nice feature. I'm actually going to show you a different record. So we're going to go to a moth record held at University of Alaska Museum of the North. So just the basic structure of a record, we've got sort of all the basic occurrence data summarized up top, and then in each of these data blocks we go into a lot more detail. And so Arctos can handle multiple identifications per record. This one has 2, so clearly it probably came in as bulk sample identified to class before being determined to species. We can check out the classification information here, and I can actually navigate to the taxonomy detail page and see all of the different taxonomy trees available from different sources via global names, so I can clone any of these. And then I can also look up top to see all the records in Arctos that have been georeferenced that have this taxon assigned, as well as media relating to the species and any synonymy. If I go next to agents, we see collector and preparator information. And so if I go to Derek Sykes' profile, he's the curator of entomology at UAM. We see his publicly available identifiers, including his ORCID ID, his name variants, as well as all the relationships of other agents to Derek. And so we see a volunteer, his family members who actually have clearly deposited collections at UAM, and then his many colleagues. And so essentially what this page does is it aggregates all of the times that Derek's name has been recorded throughout the database and pulls it into a summary page. And this is a publicly accessible page. And so now we see all of his activity. He's collected and prepared over 76,000 insects, and a few other, and some vertebrates. And so we can navigate out to those records easily. We can see all the media he's ever created that are tied to different records, as well as his identifications, his project involvement, which I'll get into in a minute, his publication involvement, and these are all linked to records. And then basically wherever his name is recorded, so any identifiers, determinations, anything he's entered, assigned, or edited, all right here. So very comprehensive summary of his contributions. Next, we see that this moth has actually been cited in a few papers, so we can easily click on that DOI and grab that paper and check that out. We can also look at the publication record within Arctos, and so if I click on this link, it'll give me some of that publication metadata, but we'll see that this moth is cited along over 4,000 other records, and this publication has been cited in 28 subsequent papers. Next are identifiers and relationships. So this moth has a barcode. It's also been sequenced, and that data lives in GenBank and Barcode of Life, so we can navigate out to those extended specimen resources, check out the sequence data, and see that reciprocal link back to Arctos. And then we see that this actually has a relationship to another record in Arctos. This is a parent of this other moth, clearly the same species. And so we see an offspring relationship pointing back to the parent, and see that it's actually 30 individuals in their first instar. So Arctos can really capture relationships, and we have a talk on this, but essentially these could be biotic, parent, siblings. It could be ecological, like eaten by host parasite, found on or in, and then any meaningful association, so maybe things that were found in the same fossil matrix or collected in the same lot can all sort of point towards one another. And then we have the publicly available transaction data, so I'll show you what's available for the succession. So there's a nice little summary of some other records in the succession from Fish and Wildlife Service. We see media associated with the succession, and we can also map these nearly 2,800 records using Berkeley Mapper. And if we click on any of these points, we can call up those records and actually navigate to those individually. So lots of different entry points to different records. I'll talk about projects in a moment, but here's some record attributes. So I just want to point out that we have a lot of controlled vocabulary in Arctos, and so if we click on any of this hypertext, it's going to give us definitions so that we're all entering our data consistently. Then we get to the place time block, and you'll see that we use Google services to underpin our locality data, so anything with coordinates will get mapped. And we also use GATM as our geographic place name authority, which allows our data to be spatial, which I'll demonstrate in a moment. I also want to show that we can actually link images and media not only to objects or records, but also to places, events, and people. And so Arctos will automatically pull this image into any record from St. Matthew Island. So that's a nice feature. And then we've got parts, so we see that this is a whole organism. We also have a DNA sequence, and that the preservation type is pinned. So next I'm actually going to log in so that we can see some of the permissions available to operators. And so I'm going to search for a herp record that has a CT scan and that's been cited in a paper. And so now I'm limited to my record set. So if I submit that query, now you probably don't notice this, but this display is a little bit different. This is my customization, so I have a nice little media preview for all my records. Oops, and I also wanted to actually show spatial search, so I'm going to do that search again. So because we use that GATM authority, I can actually just draw a polygon around northern Mexico and search that way. And therefore, you can search across multiple administrative units as well as marine localities, which can be a little bit tricky in terms of place names. So it's a handy search tool. And let's see, is this the one? Yes, and I'm going to go to this False Brook salamander record. And so now you see my banner up top. Oh, no, this is not the right record. There you go. This also has a couple of citations associated with it. we see that there's link outs to Morphosource, so we've actually scanned this individual, and so we have both the CT data and the 3D mesh hosted in Morphosource and have those link outs, and we can check out that 3D model. We'll let that load. Okay, so now their extended specimen product. So I've got dorsal ventral views, and I checked this morning. I have a ledger scan, but I think our library server is still down this morning. It was available yesterday, but we might have some issues. Yeah, okay. Well, if we've got the access to the primary data, which is nice. And now that I'm logged in, I have access to my transaction data. So you'll see that this salamander has been involved on multiple projects. So I will navigate out to the project page, and I'm going to talk about projects quite a bit in more depth in the last talk in this session, but these are pages that are publicly accessible that allow you to group records together. And so, for instance, this project page is highlighting an NSF grant that we had to CT scan and image herps from Central America and Mexico, and so we see all the many agents that have worked on this project, and many of them are in this room. We've got the abstract for this project, as well as a link out to our Morphosource page where all the CT scans are housed, a link to the OVERT project, which is the parent grant you might have heard of, the publications that have resulted so far, and a way to navigate to those individually cited specimen records, a summary of the catalog records used, and I'm logged in, so if I were logged out, it would actually show the other collaborators on this project. And then Arctos has actually gone in and pulled all the projects tied to those specimens I've identified as belonging to the OMESO project that have contributed to those specimens. So essentially all these historic expeditions to Mexico in the 50s and 60s where these were collected. And then we've gone on to digitize them. And then we have a nice media gallery here so people can check out our pretty focus stacked images. And then in our parts, now that I'm logged in, I've got access to sort of the loan information that's really delimited by parts. So you can see some of my loans are actually media loans, and then some were of the actual voucher. And so if I go to the loan, I can see all of the fields that are going to populate a report, a loan invoice, as well as have a little summary of, these 34 adults went out on a project for a morphometric of evolution associated with arboreality and plethodontid salamanders. We've got shipment information, and I'm also going to quickly demonstrate how you can actually generate a report in real time. So if I click on my loan invoice, there it is, pulling real-time data. And because we're in a shared environment, you saw all those other institutions. If I like how they format their loan invoice, I can clone it and then just kind of tailor it to my needs. This specimen is also object tracked, so to the jar level. So you see it is in a jar, on a shelf, in a case, in a room at my institution. And I can easily view who else is hanging out in this jar together, as well as see the jar metadata, which I don't have a lot populated, but just to give you a sense of all the containers that sort of hierarchically nest within one another. A lot of people, of course, use this for tissue tracking. And then we can also track environment. So there are a lot of environmental parameters like IPM traps, temperature, humidity, and anytime you're logging these, you get a timestamp. So for instance, we've got timestamp technician, the concentration of ethanol, last checked in 2018, so I need to review that. And then lastly, we have link outs to aggregators. So I think something's happening with the iDigBio API as of this morning, but we've got a link out to GBIF. Usually iDigBio appears alongside. So anything served to the IPT, you can easily explore that record and see how it's displayed in GBIF, iDigBio, as well as Globi for relationship data. And we see a couple of preservation histories. One other record I wanted to just quickly show because it has permit data. This is a bird record. You can see how, like, atomically we can track trait data. But if I go to this accession, you'll see all the permits attached to this accession, as well as explore that individual permit and see all the accessions tied to it. So really handy for tracking compliance. And then lastly, I'm just going to wrap up by saying once you're logged in, there are a lot of tools available. I'm not going to go through these, but most of these relate to being able to bulk upload or edit records across any node of the database. Super simple. And then we have a lot of summary stats and qualitative metrics available, which I'll demonstrate in the last talk, but a lot of data quality too. So here's one last report that is pretty handy that really helps you identify data inconsistencies. So this is looking for date inconsistencies. So for instance, this record says it was collected in 2017. However, this collector died in 1900. So it's pulling from agent date of birth and date of death, flagging those records so we can help improve our data. So just a cool feature. So I know that was a super fast tunnel through, but hopefully it gives you a really good idea of how highly networked this data model is and really how it can really help you manage both your digital and physical collections. So thank you.
And next up, we have Andrew Dahl from the Denver Museum of Natural Sciences. You may have to navigate yourself. Yeah, there you go.
Yeah, wow, Emily, that was amazing. So, yeah, I'm here to talk about our object tracking in a little bit more detail. My name's Andy Dahl. I work at the Denver Museum of Nature and Science, where I'm not fighting crime and bad data with Arctos. I am the collections manager for the zoology department. There's a shot of our collections facility, a little shout out to Delta and their amazing cabinets there. We have several of our collections in Arctos. Our two arthropod collections are actually in Symbiota, but we work extensively in Arctos for our other collections. My most extensive background is working with our vertebrate collections, so that's what I'll be speaking about mostly. But most of the time when I'm dealing with requests and things, the first question is, where are these things? So we are actively incorporating our material into the Arctos object tracking system. As you can see there from the abstract, a thorough and up-to-date inventory system is essential for addressing access and usage requests in a timely and efficient manner. Sorry, I don't know why I'm so nervous right now. It's been a while. I give lots of tours, and those are comfortable. But so I was just going to run through some of the basics of this object tracking system, which Emily just showed us real quickly. Key points of that being that it's hierarchical, so you can find exactly where things are in your systems, in buildings, things like that, and how it's integrated in the systems. So this is our find container page here. So you can search for your material in multiple different ways on the side. You can search for parent containers. You can search for the actual barcoded object you're looking for. You can search by labels if you want to give things names. You can type in, freezer number 2. You can put that in there and search for all that. You can search by part types. You can search by container types, so you can pull up all your cryovials. And then it displays in this hierarchical tree here. You can see this is I searched on one of our cryovials, which we have just moved all of our tissue samples from -80 freezers into this new liquid nitrogen storage facility. So this is our most thoroughly tracked collection that we have here. So I will refer to that repeatedly. So you can see we've got a vial in a freezer box, in the freezer rack, in our cryo tank, in the vault at the museum. On the right-hand side, you can see some of the details of those containers, the labels, the barcodes. There's those links to the editing the container. You can see all the collection objects that might be in a container. And you can select multiple or any of those levels on the tree. Just click the box and you'll have access to all of that material, not just the one that I searched on. So that's really handy. And then you can also see on the bottom there's that tree again, and you can click on each of those individual layers in your hierarchy, and it'll bring you to those pages. So this is that edit container page that Emily just showed us, and you can see This is where you could edit the label, change the name of the object. We generally just use our human readable version of the barcode for our lowest level containers, but as we get up, like our tanks were named Arctic Fox, Arctic Puffin. They've got nice little pictures on them in the lab. But you can put in lots of metrics on these containers. So you can see there's dimensions. You can put length, width, height, and you can use those as restrictions so you don't put the wrong type of container into other containers. So if I put in a size of this cryo vial that's bigger than the box it goes into, it would give me a warning and wouldn't let me put it in there. So that's a handy feature. There's also weights. So for those of you who may be tracking like paleontological specimens, there's issues with weights and floor loading and weight capacities of shelving units. You can put that in here so you can track and make sure you're not exceeding any of those capacities. You can put in positions. So for like freezer boxes, we have 10 by 10 boxes or 9 by 9 boxes. You can define that in here and make sure everything is going into the appropriate slot within the box. You can put in textual descriptions, remarks, and then you can also move things around from this page if you want to put it into a different parent or move everything from this box to another box. That can all be done from here. And then at the bottom, you can see we've got our container environment fields, so we can track the histories, and I'll get into that in a little bit. So yeah, you can track your tissue collections in freezers, but we also have multiple types of container trackings. There's a whole code table. Oops. Sorry. You guys can't see that, can you? All right, anyways, Emily clicked the drop down. There's a whole list of different container types that you can select. So just a few examples here. If you want to track your insect collections, you can take that down to your cabinets, drawers, unit trays, and pins. Sometimes you have multiple insects on a single pin, so they could all go and use that container type. Field collections, you can track any sort of material samples that are in your freezers, on your shelves, in your cabinets, or you can also use it for tracking exhibitry. And if that stuff moves around, you can track that through the building. So very handy feature there. These tracking systems are integrated throughout the system. So this is a snowy owl that I prepped a number of years ago. You can see on the top line there's just the data summary of what kind of parts and locations. If you were to scroll down to the bottom of the page, and I wasn't as brave as Emily to try and do it live in front of you guys, you can see our parts tables here. And on the bottom here, we've just got our tissue collections that are actually barcoded. We're working on our skins and skeletons. We're rolling that out slowly, but it's coming along. And you can see every level of that same hierarchy going from tank down to the cryo vial where those are all at. You can see the preservation types, and you could have multiple things. If it's in ethanol in the freezer, you could have multiple preservation types, and you could also track history of those preservation changes over time. It's also incorporated into our search windows. So when Emily did that search, you can use that tools drop down there, and you can pull up a parts list. So often you're not just looking for a single tube, you want 50, 100 specimens that are going out on loan. You want to find out where they all are, so you can use this. We have on the right here, once you click on that parts, it'll take you to another page, and you have 3 different options of how you could display these locations. There's this flat or legacy view. This is what I use mostly because that's what I know and I don't change easily. It's pretty basic. It's got the catalog number, the part type, the collection date, and then the lowest level hierarchy. So it displays immediately the 7 from the parts you're interested in, the 7 layers up. So you can filter that with let's see, you can filter by part type. You could select just the tissues you want or the skeletons, whatever. Yeah, that's all I'm going to say about that. This is the attribute-based merge view. So this one gives more of the collecting information. You've got all the date, the location. It also gives you attributes from the specimen. It'll give you attributes for the parts if you have those as well. So this is a very robust or very rich set of information. So it gives these really long columns. For example, those attribute column, that's a JSON string, which you can download all of these tables in a CSV format, and then you can bring it into whatever program you're more comfortable with for sorting and arranging all this stuff to help you find and chase this stuff down. This is the third option, the bulk edit part view, and this is mainly for getting those part IDs. So everything in our system, all of the parts that we input, they get a part ID so that you can track it through the system. If you want to make bulk edits to those parts, you need that number, and this is the easiest way to get a whole series of those instead of copying and pasting. Yeah. As we saw, you can track environmental histories. So currently we have the ability to track temperature, humidity, ethanol concentration, isopropanol concentration, IPM tracking, and there's just an option for checked or not checked. It's a plus or minus. Did somebody check on this thing? And it'll record the date, the person, and your values there. This is a little shot of the tools directory for the container tracking, and you can see there's lots of bulk load options, bulk loading series, creating the barcodes, creating the containers, doing lots of editing. That can all be done from that tools directory. We like doing things in bulk instead of one at a time. If you don't have barcodes, you can still use the system to help you track your material. If you can use those names or labels for inputting the stuff in the system and keeping track of what you got. This is really best used for temporary or consumable objects, things that you just it's waiting to get cataloged, accessioned, whatever, and then you'll input it into the thorough system. The one caveat here is that it's not hierarchical without the barcode. You can't track it through the system, so if you really need that, reserve a barcode series and just input those in there. It's not very difficult to actually get that set up in the computer. It might be a little more work to get stickers on things or whatever, but it's definitely gives you that hierarchical thing. But you can record that into remarks and things, so you can still find these. Some additional considerations: what kind of barcode series or barcodes you want to use. It's a shared system, so all of us, we have to reserve whatever type of barcode series we want to use. So you can see in that line there, that's one of the Denver ones. We put in the DMNS prefix to make it unique to us, and then we've just got random character series there. Some collections have wanted to use or do use catalog numbers as their barcodes. That works well unless you have multiple part objects. You want to be able to track each part individually. So generally, we recommend these kind of random series. You can reassign barcodes. If a sticker falls off, just stick a new one on there. It doesn't really matter. It doesn't mean anything, but it's easier to track. Another thing is creating positions in kind of the higher level containers. Something to think about. Those are kind of harder to change once you get it established. So it's not impossible. You have to remove things and then adjust it and then put things back in if you want to do that. But it's best to try and figure out how you want things set up as best you can before you create any of that structure. And then I mentioned the part IDs. Those are full hyperlinks, so you can share those with people. And if I were to click that link and it actually came up there, which I don't think it's going to, it would go to the specimen record and go down to that parts list and highlight the one part that you actually use. So you could technically cite specific parts in different papers or projects and know exactly which one of the 10, 15 parts you have with that record that you're talking about. And that's about it. Yeah, we've got our NSF grants. I want to acknowledge the member institutions that really have contributed to the development of Arctos and all of the data that's in there, and of course our programming staff. So, thank you.
Thank you very much. All right, so now we are going to hear 5 minutes from Mariel Campbell, who is, let's see if I mean,
The cone, like the traffic cone, click on that on the bottom.
traffic cone. Oh, yes, there we go. Traffic cone. Okay. Yeah. And do we have oh, yeah, here we go. Okay. So we're going to hear now about building strong relationships in Arctos to extend the specimen. This will be given by Mariel Campbell from the Museum of Southwestern Biology. She couldn't be here today. And then Vicki has kindly agreed to step up and give us a demo, semi-live demo of to exemplify the concepts that Mariel will be talking about. Sorry, I couldn't find my mouse.
Hello everyone, this is Mariel Campbell. Thank you for being here today. This presentation, Building Strong Relationships, the Extended Specimen in Arctos, will discuss how Arctos enables the extended specimen. I will provide an overview, and Vicki Zhuang will follow with examples from Arctos collections. The extended specimen concept, introduced by Webster in 2017 and expanded by Lindemer et al. In 2020, refers to integrating museum specimens with their extended data and relationships, and with their downstream usage and research. The extended specimen concept starts with physical specimens in museum collections. The first extension of these physical objects is their digital specimen record and associated media, including specimen images, scans, and audio files. The secondary level of extension includes field notes and media documenting the specimen collection and environment, as well as derived data such as gene sequences, measurements, and isotopic signatures. And the final extension of these specimens represents broader data such as species interactions, traits, distributions, phylogenies, and conservation status. This level of information typically would come from publications, reports, and other research products, frequently not linked to the original physical specimens. The extended specimen concept was further expanded by Hardesty et al. 2022 as the digital extended specimen, which is enabled through persistent digital identifiers, PIDs, linking physical specimens to their derivative data. As a web-based CMS, Arctos fully supports the digital extended specimen. Arctos automatically generates persistent identifiers as URLs for physical specimens and associated data. Physical specimens are identified by a globally unique identifier consisting of a URL incorporating the Arctos institution, collection code, and catalog number. This URL or GUID creates a web-based link for every physical specimen. Arctos identifiers can then be used to create reciprocal relationships between Arctos specimens and data within Arctos collections and to specimens and data held in external collections, repositories, and aggregators. In addition to physical specimen persistent identifiers, Arctos assigns URL identifiers to all components of the Arctos ecosystem. These include localities, events, media, agents, including collectors, preparators, and organizations, attributes and trait terms and code tables, part identifiers, material sample IDs, taxonomic names and classifications, publications, projects, and transactions including accessions, permits, and loans. These identifiers link objects and tables within Arctos, but many of these are available as persistent identifiers which can be used to link to external web-based resources. So how does Arctos support the extended specimen? Since its inception, Arctos has incorporated the extended specimen network concept through web-based relationships, shared URLs, and links between, within, and across Arctos collections and to external data repositories, displaying and making accessible everything that is known about an object and its relationships, interactions, or derivatives. The use of URLs as persistent identifiers enables extension of physical specimens and all associated digital data from Arctos collections to any web-based platform, promoting the use of museum collections for interdisciplinary research. Thank you very much. I will now hand this over to Vicki Shuang, who will provide a demo with some examples from Arctos collections. Have a great day.
Thanks, Mariel. So I wanted to talk a little bit about myself also. I'm a collections manager of 8 different types of collections across 600,000 specimens in El Paso, Texas. I am trained formally in one, that is herpetology. So Arctos recognizes that relationships allow us to go beyond controlled vocabularies. It recognizes that the different data types in different parts of the ecosystem are universes on their own. And this allows me to actually improve the data quality because I don't know if the taxon is spelled correctly or the locality or anything else. So I wanted to talk a little bit also, since Mariel talked about a part of the research aspect of relationships, but also that it allows me to be a better collections manager. Emily talked a little bit about how you can get to all kinds of different parts of the ecosystem through our relationships and our links. So if I have specimens, especially because our curators are traditional naturalists who love everything, so they collect everything, and so they're not just collecting reptiles and amphibians, but shells and fossils and all sorts of things, is the same person that is recorded in our fossil collections, the same person in our herp collections. And I can look at that through the histories of that particular person, which records even after they have passed away. Richard is not dead, by the way. But when we continue to discover things through these relationships, we continue to add on to his aspects. And even his students and their grand students can be recorded through this because you can just dive into a deep rabbit hole. This allows me as a collections manager, especially if I'm entering a collection, to understand the history of all parts of our collections. It's a great way for students to also understand where our collections come from, the people that are connected to it. So it's not just collection management of objects, it's a collection management of a multiverse. So to talk about a little bit of the relationships act on a single means. So the same way that we connect to external relationships is the exact same way that we do internal relationships. Internal relationships, though, have this advantage of having reciprocal relationships. So when I came into the collections, I found that there was a separate osteological catalog as our herp catalog, which is extremely annoying to me, especially because that means that the data has been transcribed twice. That means that I don't know if the locality information is the same recorded in the osteological collection as the regular herp collections. But because we recognize that localities are a linked universe on their own, I can quickly check it because it should hopefully say that it has 2 objects or records connected to it, which I have a feeling that it's not because, data transcription errors. But because we have explicit relationships, so you can see in this record, I can then record that the same individual is in our herp catalog. It can go into external. So if I transfer my part to another institution, then that will also be reciprocated back, and it allows me to track where the hell that thing came from. So while this helps me in collections management, it's also a really great research tool. And so to show you a little bit of that, I don't know if you've ever gotten those research requests. It's like, do you have any animals that were specifically eaten by a particular snake in a different country? In this case, we have anything that was parasitized by flies, and then also detected for hantavirus in Panama. And so, yes, that actually does produce a record if you ever have a very specific, complex question from a researcher like that, which one day there will be. And this is a really, let me just sign out right here. Yeah.
That was an example of the collection levels permission.
Yes.
I can't see.
Everything is on purpose.
That's a feature, not a bug.
So you can see the record here, and that this allows you to have these really cool relationships. So here it says the Museum of Southwest Biology. It is the this host is a something. Thank you.
This is the community of practice.
Exactly. I rely on our community to actually know these things. And that is actually a really great point, too, because then anything that I build is used by the rest of our communities and also our external communities as well. And so that really also helps our data quality. And so from this particular link, I can look at also the actual parasite, which is Diptera, which is what our search runs on. This is really neat. This is just a single record, right? But this also can go into all sorts of different directions. And as you can see, they operate off of this single or code table that can also be detected for hantavirus. And I want to pull these together. But you can see that there's all sorts of different relationships, as Emily has said. And if there's other relationships that you're really interested in, you can always talk to us, and we can always add those relationships, because we love giving data structure, because that means being able to discover data from multiple angles, because you always have lots of different researchers that can come into specimens from different perspectives, and we want to be able to enable those perspectives. And given that we've had some discussions from other talks about like absence data, right? So we're able to also enable that by saying that the specimen was examined, which if I can find that somewhere, I cannot find that somewhere. Oops, I picked up the wrong code table. But we do have a examined as a controlled vocabulary that allows you to say, we did look for something, but it didn't actually exist. So that actually enables our absence data. I think I have one more time, a little bit of time to showcase the really neat way that we can look at the structure of this data. So we, just running a very simple search on how many parasite relationships that we have, there's over 42,000 records already, thanks mostly also to Parasite Tracker, but we still have lots of collections that continue to add parasite data, which is super cool, because now we can ask all sorts of questions because we have this data structure. So we can look at parasite relationships over time, have hosts changed over time and space, and because we have this really neat tool in Berkeley Mapper, if it will load, which I actually didn't load this. Oops. Yeah, exactly. So this is all the parasite relationships mapped over space. It'd be super cool to add a time scale to this thing. And thanks to the SABI grant, we were able to do super awesome things like this spatial intersection, which allows us to even look at data across biomes like that. So you can see that most of our parasite relationships in the database right now, there's about 12,000 records in temperate forests and 6,000 in boreal, and you can ask all sorts of really neat interaction questions. And then lastly, I wanted to give a plug to our entities. So we've talked about relationships across like a specifically formal structured relationships feature, but we also have entities that if you are cataloging separate objects over lots of different collecting events or even across institutions, Arctos entities allows you to aggregate all of those records together. This is for a zoo animal at the Albuquerque Biopark named Kianka, and she has been donating blood samples across several time points. So there she is. She hasn't moved very much. But it does allow you to map a animal across space and time if you're encountering that animal multiple times. So yeah, I just want to leave one more slide up. But yeah, relationships make the Arctos-verse a universe. It bridges our nodes of the Arctos system, and it keeps building out over time, and it will continue forever. It enables us to have better collections management and build on new innovative research tools, and we continue to find new ways of an ever-expanding universe.
Thank you very much. That was quite a journey there. Okay, so next up we have Hannah Appia-Madsen, who will be telling us about one of the newest collection types in Arctos, or at least in the last 3 years. So
Sure, yeah.
take it away. Thank you.
Is it doing the things? Ooh, fancy. Hi, my name is Hannah. I'm here from the Ocean Genome Legacy Center. We're a marine genomic resource collection, and I'm going to talk a little bit about our special collection and particularly how it functions in Arctos. So a little bit of brief history. We're about 20 years old, and in our 20-year history, we exclusively used a FileMaker Pro database. And that's because when we were founded, other collection management systems weren't well positioned to handle genomic data. But now genomic resource collections are becoming more of a thing. So we joined Arctos in 2023. And as when all collections join Arctos, they're special in their own ways. And so we were the first dedicated marine and dedicated genomic resource collection, which means that we needed to work with the community to make some positive changes. And so wonderfully, because Arctos members get so involved in the community, I'm going to talk to you today about 3 particular areas that we made some changes. So we talk about marine geography, marine event attributes, and genomic part attributes. So a very brief background on what OGL is. We're a genomic resource repository. You can read a paper on the left, more if you want to know about our deal. We were founded by Don Combe about 20 years ago after he read an op-ed in Science in the year 2000 about why we should all be creating DNA banks for endangered animal species. And that's that paper on the right. And so he decided, yes, we're going to do this for marine ecosystems. We're located north of Boston. Oh, I love how excellent my fonts are. All right. We're located north of Boston, about 20 miles, on an almost island. And that's a picture of we're at Northeastern University. That's a picture of our Marine Science Center. We've got a lot of really excellent water views. It's delightful. Why do we focus on marine life? Well, this is a map that I pulled from GBIF earlier this month, and you can see that the gaping black holes of nothingness really nicely overlay with our marine ecosystems. So we have a lot of information about our marine ecosystems that we don't know about. And also, our marine ecosystems are affected very particularly by climate change. This on the left here is a map of sea surface temperature changes in the last 100 years, and our oceans have already experienced more than 4 degrees Fahrenheit increases in sea surface temperatures on average, right? And then oceans get this extra special ocean acidification thing going on in them where atmospheric CO2 goes up, and so seawater CO2 goes up, and that means the pH drops. And for anything that has a calcium carbonate shell, think mussels, clams, corals, their skeletons basically dissolve into the ocean. So real bad, right? And then humans do a lot of other direct things, like we put a lot of plastic inputs, we spill oil, we overfish. We're having all of these huge impacts on our marine ecosystems. And that means that there are predictions of lots of biodiversity loss. And this is a map where red is even more bad than blue, but all of it is bad, right? This is all local endemic biodiversity loss in our marine ecosystems. But why do we focus on genomes? And that's primarily because they're very physically tiny. So we have a very different footprint than perhaps a wet collection would. But they're informatically very large, so we can learn a lot of information about our ecosystems, about species, about diversity, even though we have very tiny samples. And then also they're generally speaking very uniform. So we can treat the DNA from a bird the same way that we would treat the DNA from a flatworm, right? They're like taxonomically very diverse, but we can treat them uniformly in the collection. And so just like very briefly, our collection is about 35,000 records from 5,000 different places, and depth ranges from 0 to almost 6,000 meters below the surface of the sea. And we have about 5,000 taxa. We're mostly marine invertebrates, but we also have teleost fish well represented. Okay, so pivoting back to Arctos. So the first thing I want to talk about is marine geography. Marine geography is substantially less well-defined than terrestrial geography, but it's really valuable to have both terrestrial and marine boundaries connected to our records. And that's because it facilitates, for me, that's because that facilitates permit management and is also a really important search parameter. And so for those of you who use Arctos, if you'll excuse my very simplified representation of the database, Arctos starts with geography on the far left. And when we started this work, geography was either based on GATM if it was a terrestrial system, or IHO if it was a marine locality. And then a single geography can, of course, have many locations. And then a single location can have many events, and then you can, of course, collect many objects at a single event, at a single location, at a single geography. So with some example data, we're going to go exactly the opposite. I collected some mussels, some sea stars, and some snails by hand, by scuba, or by snorkel at these two beaches that are in the Atlantic Ocean. But are they in the Atlantic Ocean? We ask this question all of the time, right? That is one of those beaches that I just mentioned, and is it in Nahant, Massachusetts, or the Atlantic Ocean? And the answer is, it depends on the time of the day, right? If it is high tide, it is unequivocally in the water. And if it is low tide, it may be extremely dry and very easily Nahant. But we need to be able to ascribe both of these attributes to this particular location, because keeping track of the time of day is nonsense in this context. And so we worked collaboratively with the geography working group, shout out to Jenna, to like sort of loosen up the way that Arctos dealt with geography, such that we could have both terrestrial and marine values associated with a specific locality. So you can see here that this place here around Nahant, we are calling it the Atlantic Ocean and the Gulf of Maine, but also the US, Massachusetts, and Essex County. So that's like that is very important for us, because it means that now we in the public can search for records in Massachusetts and get records that are both on land as well as those that are just offshore. So before we made this change, if we had searched for Massachusetts, we only would have gotten those blue records at the top. But now, because of this modified locality, we also get the 400 records at the bottom. So that has a really big impact for us and the way that we can manage the collection. Okay, so for marine event attributes, marine data are also often organized in different ways than terrestrial data. We care about things like vessel names, remotely operated vehicles, or autonomous underwater vehicles, cruise numbers, dive numbers, all that sort of thing. And if we go back to that GBIF map from the beginning, you can see really clearly that a lot of our marine data comes from very clear cruises. That's like a very clear line of a ship going back and forth and collecting information. And you can see that across the Atlantic and down to Antarctica, like we work in cruises. And so we needed some more marine-specific event attributes, right? We wanted that more structured, code table-based, can't make mistakes, can make fewer mistakes kind of data management. And so for this, we worked closely with Teresa Mayfield-Meyer, and this is a particular collecting event. At the top of the record, you can see when it happened, hooray, and some verbatim information about the event. But then we have very structured in these attributes at the bottom, the cruise, the dive, and the vessel information, which which means that we can very clearly search in a very specific, structured way to get this information. And this is, again, amazing because now we and the public can search for records associated with specific vessels, cruises, or dives. I love how this looks. So the important thing to take away here is now I've done a search on my collection for the research vessel Okeanos Explorer, and I found all of the records associated to that with that particular boat. Or vessel, a ship, whatever. And then I can go more superb. I can go even more. I can like zoom in a little bit and say like, hey, I want to know about those specific records with a very specific cruise, and even the 9 records that are associated with that particular dive when that ROV went down and came back up and collected these things. And so that's really wonderful for us. It reflects the way that researchers think about marine data, and it means that it's easier for us to organize it and make sure that everyone is putting the information in the right places. Okay, now I'm going to pivot to the genomic aspects of the work that we did with Arctos to talk about some genomic part attributes. So a gold standard for a genomic sample includes a lot of genomic-specific attributes, and for those of you who are at the plenary, this may not surprise you, as we heard that genomic information is fun. And so we want to know things about preservation methods, extraction methods, quality metrics. We have a lot of kinds of enriched data about genomic samples. And if we return to my very simplified representation of Arctos, we have a record that can have many, many parts. For us, that's primarily tissues, DNA, and RNA. And then those parts can have attributes. And before our work, our options were really to have preservation, volume, and weight were the kinds of things that we could describe. And so, for instance, here we have Mytilus edulis, a blue mussel. I have some gill tissue. I have some DNA. I can say that the gill tissue is 50 milligrams and is in 70% ethanol. And I can say that we have 200 micrograms of DNA. That's all really wonderful and lovely, but I would like some more genomic-specific attributes that I can report in a really consistent way that researchers can search on. And so, again, we worked with Teresa Mayfield-Meyer to add these attributes to parts. And this one is adding different kinds of preservation methods so that we can say that our DNA is in buffer AE, which is a really common elution buffer if you use a QIAGEN kit. We have extraction methods where we can specify the specific kind of kit that we used in order to do the extraction. That means that researchers really immediately understand what kind of protocol happened here. And then we have a variety of quality metrics. So we have things related to DNA purity, 260-280 ratios, 260-230 ratios. We also have information about the concentration of DNA and about the quality of the DNA in terms of like what the molecular weight is of that DNA sample. And that's so important because now researchers can come to the collection and have access to the information that they need to evaluate the appropriateness of our materials for their applications, right? A thing that I've heard repeatedly in talks here is like, you don't want to make assumptions about what researchers want to do or want to know about your data. So this way, we don't have to make any assumptions. We can say, here's all of the information, you decide if it's appropriate for the kind of sequencing that you want to do. So in conclusion, through collaboration, emphasis on the collaboration, we have added marine geography, marine event attributes, and genomic part attributes to Arctos. Hopefully I've demonstrated how the flexibility of Arctos can be a huge benefit to less typical collections. And now Arctos is better positioned to welcome marine and or genomic resource collections to the fold. And so I'm going to end with my very ridiculous pitch that, like, yes, Arctos may cost money, but the collections work in community is truly priceless. And as a tiny collection of 3 people, the fact that we are now a part of this huge community has been literally life-changing. And so with that, I would like to thank these very specific people, Teresa Mayfield-Meyer, love of my life, Jenna and Phyllis, our marine comrades, Marielle, our fellow genomic resource collection champion, Dusty for all of his developing brains, Michelle, our fearless leader, and everyone who is in the working in issues groups who contributes to this really vibrant conversation that we get to be a part of now. I'd also like to thank all of our undergraduate students who have been with us as we transition to Arctos. It's been a challenge for them, and so they've been providing us with a lot of feedback, and that's really wonderful. And then I'd also like to thank the Gillette Charitable Lead Trust, who has funded our transition to the Arctos database. And I will take any questions at the end in the panel section. Thank you. Thanks.
That was wonderful. So we have one more talk now. Emily Breaker's coming back, and she's going to talk about impact tracking, and then we're going to have lots of time for discussions and hearing more ideas. Hopefully, this is. Whoops, I almost had your talk going here. There we go.
All right, hi again. All right, so tracking collections metadata and curatorial effort is central to communicating our activities with stakeholders, and the collections community really relies on a collections management system to summarize these metrics for inclusions in things like grants, reports, audits, and inventories. But more and more, justifying our relevance to stakeholders is a reality, and therefore higher level impact tracking is essential for the future of museums. So this talk is going to provide a brief overview of all the Arctos tools that aid in this endeavor. And a lot of these functions really go beyond basic tallies to really leverage Arctos's highly networked data model to provide really comprehensive summaries of impacts and effort. So our most basic tool is known as System Stats. This is a table that just provides a lot of at-a-glance statistics that are really useful for reporting. And so it will summarize things like number of catalog records and taxa, media and loan counts, as well as extended specimen resource tracking like publications and citations, as well as data products that are linked with Arctos records like GenBank sequences. And this table gets archived monthly so that managers can really monitor growth over time. We've also recently partnered with grad students at UC Davis to develop a Tableau dashboard, and that's pulling in those system stats metrics. And you can filter it by institution and collection. And so these provide really dynamic visualizations, but they're also really easy to screen grab and include in reports. And so our next tool is Arctos Projects, and this is a really unique feature because it helps create a multidimensional picture of the impacts of collections. And so these are publicly accessible web pages within Arctos, where data managers can group records based on a thematic collection. And so what they do is they help track activities. So this could be things like a research project, it could be a grant award, an expedition, really any meaningful assemblage of records that you want to track. And then on the web page, you can include a project description, identify the people involved, link out to external resources. And then these pages will link to the direct products of the activity. So it will be either the catalog records that you used or were contributed by the project, and media or publications that were generated by the activity. And then once you link records to a project, Arctos is going to automatically pull in those secondary products that are tied to it. And so it's going to pull in all those existing publications and projects tied to the objects, as well as keep track of all the future products that are generated. And this really illustrates how projects are connected to and support one another through time. So in this example from MVZ, we see a project page that highlights the Grinnell survey in Yosemite National Park. So we've got the dates up top, the agents involved, a project description, and then a list of all the catalog records that were collected on this transect. And so it's about 7,000 specimens that were deposited at MVZ. We see that there were 2 resulting historic publications, as well as a media gallery. So there's historic site photos, as well as images of the animals in life. And then we see that this page finally highlights the 37 projects that have gone on to use, I can't read this slide, to use those specimens that were collected on the transect, producing 15 publications and over 13,000 specimen citations. And so this is the project page I demoed in the preview, but this is, again, our digitization grant. And this is a page that I've included in our annual NSF report to really help track our products. So again, it's got all of our project technicians that we've trained in STEM techniques, our products, including focus stacked images, CT scans, and resulting publications, all gathered into a single gallery. So rather than our funders or our end users having to query this all through the search portal, it's just there in one place, easily summarized. And then this is a multi-institutional effort, so you'll see that there are actually catalog records that are sourced from 3 different institutions, all digitally united on the page, and then all the linked projects. And so all the multiple historic expeditions that contributed those records that we digitized for this project, and includes the Grinnell survey, so a little tie back. But all those project pages, of course, link to their own records, their own projects, and their own publications. And I didn't have to manually create all these dependencies in Arctos. Once I identified the group of records, Arctos, the structure, facilitated that compilation. This is another use case where project data was pulled into the UTEP's annual report, so the annual report's on the right there. This involved a grant that incorporated a large invertebrate collection that was digitized. And so rather than having to spend a lot of time tracking down these sort of growing statistics that are changing every year, all this information is really readily accessible on that project page, easy to pull into a project template or share that URL with stakeholders. And then now looking at a really well-established project, we really can see how powerful this tool can be and the downstream impacts. So Beringian Coevolution Project was a series of expeditions to the Arctic to collect mammals and examine them for parasites. And this project examined over 1,200 voucher specimens, as well as added nearly 30,000 new specimens to the various collections listed there. And thus far, 216 publications have been produced, citing over 8,000 specimens, and all the papers are directly linked to the project page, and so people can click on those DOIs and explore the papers, as well as navigate directly to those cited specimens. But the magic really happens with the automation. So we see up top there are 20 projects that contributed specimens, as well as 157 projects that have gone on to utilize the specimens collected on these expeditions, ultimately producing almost 300 publications citing over 69,000 specimens. So mind blown. This really highlights the incredible productivity cascade and really shows how projects can provide a really robust infrastructure for deep tracking research products and effort. And so projects showcase the utility of collections through time. And we can, of course, do this on an individual record basis in Arctos, but projects really allow us to take a big picture overview to show how collections and the people and products that are tied to them are all sort of interconnected. And these are just some other project examples to give you a sense of the diversity of thematic groupings. And I want to point out that a lot of projects are retroactively assigned by data managers. So for instance, this National Park System project tied to my collections, all of the records linked to it really represent multiple field expeditions headed by different people in different decades on different NPS properties. However, they are related, right? And so it's a way to bring these all together and really illustrate how those public land specimens have been incorporated into research and really capture our own institutional effort through time, as well as just provide a really simple, elegant way to communicate that with our agency partners, right? Just sharing the link. And then one other feature to highlight specifically for funded work is that Arctos will calculate the total value of connected projects. So in this example, MVZ received a grant for nearly $500,000 to ingest an orphan collection and then process over 27,000 backlog specimens. And basically this effort went on to support later projects that use those specimens in different projects, and they all had associated grants valued at over $3 million. And then, of course, some of those projects went on to support other funded work. So even though it's not a perfect proxy in terms of how much one project has contributed to another, it's still a really compelling statistic for stakeholders in terms of understanding how research dollars compound and multiply and contribute to an ecosystem of supported work. Publications are, of course, a metric that we all like to track, and it's very easy to do this in Arctos. So in a similar fashion to projects, Arctos is really able to demonstrate how publications have gone on to support other research. So we have an integrated Crossref tool, and so Crossref is using the DOI to pull in real-time data in order to display how many times a work has been referenced by subsequent papers to really get at conveying productivity and downstream impacts. And like projects, publications are shared across Arctos, meaning that a publication record can contain citations from multiple institutions, like this one. And so it's a really nice way to highlight collaborations, as well as have end users navigate to all those cited records regardless of what institution or collection they're deposited in. And then beyond tracking collection metrics, agent activity is another way to showcase effort and impacts tied to people that are affiliated with collections. This is kind of the constellation of data that's collated within an agent profile, as we saw. So these data are automatically pulled in, again, by wherever that agent name is recorded in the database. And so again, you don't have to manually create all those linkages. So this is an example of Arthur Harris, who's a paleontologist and director of the UTEP Biodiversity Collections. And I know you can't read that, but it's really there to show the sheer volume and diversity of data that gets assembled for any single individual. And of course, all those blue hyperlinks are linking out to records and different tables throughout the database. And so users can really easily view a synthesis of his work. They can look at, his little profile blurb up top. They can link out to his wiki data, see all of his relationships to other agents in the database of the many students he's advised. His spouse is on here, who also deposited collections. All the specimens he's either collected or prepared, his attribute determinations and identifications, as well as publication and project involvement. And so agent activity really provides a holistic view of career-long contributions. And again, these pages are accessible, and so they can be shared as URLs or included in people's CVs and resumes, which is really handy for students and volunteers working in collections. And this is an example of a more curatorial agent record using co-author Vicky Zhuang's profile. And so this is really useful for collection managers in terms of tracking and communicating our impacts to administrators, especially for people who might be in temporary or soft-funded positions who might need to have to advocate for continued funding. And so you'll see that her profile highlights all the students that she's trained and supervised, nearly 100,000 records that she has entered, 33,000 media records she's created, and literally thousands of edits to different tables throughout Arctos, as well as her permit and transaction activity. So it's really comprehensive. And I do want to point out that it's also really useful to reference agent pages of students and volunteers to track their effort, because if you're writing a recommendation letter, it really helps jog your memory, like what projects were they working on, and it also helps you provide concrete statistics in terms of their database actions, so maybe all the georeferences they assigned or the number of specimens they prepared. And then agent profiles are shared across Arctos, and this really allows us to understand our impacts in aggregate. So metadata and agent profile pages can be collaboratively edited and improved upon. So in this example, this is a logged in view, but 3 different operators at 3 different institutions contributed different pieces of data to this profile. And that means in Arctos, we can share knowledge rather than working with partitioned data, doing redundant research, and creating multiple profiles of the same person. And we also have a mechanism to notify each other of a potential duplicate name and suggest merging profiles, and that really ensures that impacts and efforts are attributed to a single individual rather than dispersed under multiple name variants. And we've kind of looked at records, but this is just showing how that impact and activity data kind of sits alongside the basic occurrence and object data. And just finally, I want to point out that there are a lot of different tools in terms of data improvement that really detect gaps in our data and ultimately help our ability to track our impact. So for instance, we have bots that crawl GenBank and Morphosource to look for data that relates to Arctos records that's kind of floating out there in those other repos that managers might not be aware of. As well as a lot of reports that flag for potential data consistency and connectivity issues so that managers can resolve those, and those of course feed into impacts. So just to wrap up, Arctos can help you communicate why the work we do is impactful, and this functionality really does rely on highly standardized network data to achieve this holistic view. So we hope that you can leverage these tools for your, to demonstrate the continued value of collections. And with that, I'd like to thank our funders and the Arctos community.
Great. Well, thank you to all of our speakers. Hopefully that was a good demonstration of the Arctos-verse and what we're always talking about when we talk about community and database platform. So now I would like to open it up to discussion and questions. So first of all, are there any questions for any of our speakers? We have Nikki here. also a former MVZ Arctos user. So, is that a hand up or just a shout out? Just a happy hands? Okay, just happy hands. All right. Any questions for any speakers? Yes, Carol? Oh, sorry. Go ahead. Yeah.
Not something that was covered, but as a potential user, what is the ease of taking whatever you have, Excel, whatever, and putting it into Arctos?
Well, it really depends on where you're coming from. Sorry, it really depends on where you're coming from. I'll address that, but we can also ask some of the other new users coming in. I'll just say that wherever you're coming from, we have a discussion first of all. So we actually have data migration specialists on the Arctos staff, and that's their primary job, is to make that transition as easy as possible. We also recognize that, you know, you're going to need training. With any new piece of software, right? It doesn't matter if it's a desktop tool or a cloud-based tool. So, and we're always learning new tricks as well. So that's a conversation that we start to have from the very beginning of when we're signing the MOU to when we get you in contact with a specific data migration specialist. Right now, we have Erica Krimmel, who's well known by many of you, and she's also a deep informatics specialist, so you can also ask her lots of other kind of related questions about managing your data. So that's what I would say. I mean, so I can't answer that specifically, but that's usually our paths. We don't let you founder. So I don't know does anybody else want to add to that?
I will answer. I mean, one thing that our database relies on is really structured data. So I do think probably in comparison to other CMSs, realistically, it's a bit more work because we have a lot of controlled vocabulary, so you do have to do a lot of data normalization, and ideally you are sort of deduplicating your agent names, checking against what we have existing in the database, but it's really, you know, you see the benefits of that, so it might be a bit more work up front than just being able to load lots of maybe dirty records in, but it's a lot of data cleaning. But yeah, I think if you have pretty atomic fields and things like that, hopefully it's just kind of agents, taxonomy, and geography that are kind of the biggest lifts.
I just wanted to say that it's worth it. And it's easier if all of your staff don't go on parental leave while you're in the middle of the process. What else did I want to say? The no one is left floundering and dying on their own part is like, I cannot tell you how many like panic GitHub issues. We're like, oh my God, this isn't working. And someone's like responding within 5 minutes. And we're like, why are you up at 7 a.m. responding to GitHub issues? And yet here we are.
I felt your...
The panic is felt through the Arctos-verse. Yeah.
And we're all connected somehow. Any other, sorry, Carol, you had a question?
Okay. Mine's very specific. Sorry, the totally other end of the extreme. Andy, on the container edits, you were saying, I didn't realize you could do this. You can add labels and have containers that don't have barcodes, but then they're just sort of existing in free form, or can they exist in a room or no? Like I don't know if you can show us that or just tell me. Yeah.
No, they're kind of individual, like just kind of label markers. You can add that additional, remarks and descriptions and things that can contain that information. So it's not as functional, but it can get you started for like tracking what you have. Yeah.
I see. Okay.
Any other questions? Because if I don't hear another question, I'm going to start asking you guys questions. I actually have a set here of questions. Like I said, I want this. Like I said, I want. Oh, yes, go ahead, please. Oh, please. Yeah. Like I said, this is a open discussion time.
Yeah, so this is great. This is fantastic.
my comment is on tracking agent activity. One thing that students and early career professionals should consider is looking at their own profile and putting their contributions on their CV. So I have a section on my CV of what museums I've contributed to and in what ways. And so it's also really great for just tracking your own work, or if you have, performance reviews at work, you can say, these are the things I've done, the contributions I've made. So yeah, it's a really cool feature.
That's a good tip. All right, any other comments, tips, ideas? Before I start pestering, if anybody knows me, they know I also ask a lot of questions. Yes, oh, more questions.
This actually relates to what you just said. The question is, how if the museum is not in the Arctos-verse, because I've contributed samples and vouchers to tons of other museums.
Yes.
I was just curious how that works within the Arctos-verse.
Yeah, so if you have contributed to a museum that's a member of the Arctos, you have already started an agent profile, and we have already started tracking your contributions. So yes. So it's actually kind of fun then when you join Arctos, because, you got a lot of stuff already started to track there. You can look up other people's names. They don't have to be actually in an Arctos member museum. If they contributed, you'll probably find us on the agent page. You'll find yourself on the agent page and what loans or specimens you've contributed, and what maybe a little blurb. So we have a lot of historic legacy people tracked in that way too, and that's been really helpful. Okay, that's it.
Hi. So I want to say something that it's related to Arctos and to extended specimen discussion last time. Yesterday we had. So we are here representing Plazi and the DeSantis roadmap, and we think that this idea, it fits perfectly in the idea of the fair data and fair publications, and the way you are looking for tracing data, and you have the metadata associated to all the data, and then you can justify to stakeholders the how big is your collection. So you can do that more easily if you have all your data, the publication data online on this verification mode we are proposing. So Laurence here will talk about the roadmap a little bit more, but if you are also talking about dashboards and the way you can show your data. So now we have the opportunity to have the hosted portals in GBIF, so you can have a specific portal for your institution in GBIF. You have all the information from your institution there. And then we are also inviting you to the discussion that we will have now in the other section after the break about the fair data integration. So, Laurence.
Yeah, thanks. I wanted just to ask you a specific question about publication and how you link that. So I have seen that several times you mentioned publication, and some of them do not have DOI. So it's very difficult to track. Have you imagined the fact that we could put a PDF of that collection, put a DOI on it, and store it in BHL? If it's not in BHL, it could be in Zenodo or any other thing, and track your data from the DOI and from the publication itself.
Yeah, does anybody want to, I can speak a few words and then, yeah, okay. So, our publications predated DOIs, cross-ref.
Yeah,
So,
for
yeah,
sure.
yeah,
For
so it definitely, we are definitely looking and have imagined improving that access. I had actually seen your presentation yesterday, I think, so
Yeah.
I was definitely very intrigued. And the fair data principles are things that we keep in mind. Those are actually part of, our considerations when we're developing new tools. So I would be really interested to find out more and figure out a way that we can enhance, both our tracking internally, but also externally. So we have reached out a little bit to other publishers and tried to start, some conversations about how we could do that. I actually hadn't thought about creating the data sets themselves as a trackable object, digital object, but that makes sense, and it definitely falls
So
in place.
let's talk about that
Yes.
and deepen the conversation in the lunch.
Exactly, yes.
I'll just add too that you can absolutely tie media to our publication records. I mean, there's issues maybe if something's not in the public domain with copyright, but if it, okay, well, yeah, so you could absolutely do that. And then we support link out. So if it's available online, a lot of BHL might have a persistent ID or something, and so you can reference it that way as well.
Yeah, because it's a little bit different. There is a difference between open data and fair data.
Yes.
And this is something that we have to discuss, because a PDF can be open, but when we are talking about fair data, we are talking
Have to
about
be
giving
accessibility.
Yeah, accessibility to the all and PIDs to material citation, to the treatment, to the table, to the figure, and then all this is to the specimen. So all this is linked. And this is different, and this is powerful.
Yeah, I should also mention that we highlighted a lot of our dynamic links. We also have a methodology to create a DOI, it's an ARC, for archive data sets. That means it's a snapshot, doesn't change. This is the static. This is the static data set that was used for a particular purpose. It could be just, a data loan. It could be an actual loan of physical specimens, but it's basically a snapshot of the data as it stood at the time of use, as opposed to a lot of the dynamic links, because that was also a need that we saw. So I think we definitely have the components for creating that. We just need to get a little bit organized and collaborate. Yeah, thank you. I have it written down.
This is another potential user question. Thank you to all the speakers. This has been fantastic, and it's amazing to see how easy it is to keep everything interconnected. For records that, for whatever reason, need to not be publicly viewable, thinking of, for example, cultural collections that fall under NAGPRA, how easy is that to keep those separate, essentially?
Yeah, very, very easy now. That is a strong need for our cultural collections, but also our paleo collections. So they have a oftentimes mandated need to keep specific paleontological collection sites hidden, but they want to have at least some public accessible or higher geography available. So we allow for many of our attributes to be either public or only private according to collection access. So that goes for a lot of the locality attributes. So you can have an entire locality stack kept private just for your paleo collection, for instance, and then have a second locality that's completely public, and maybe that's only to the county level with a little remark that says, contact the collection for more information. Same thing with a lot of the attributes. We didn't highlight, since this is Spnhc, we didn't highlight a lot of the cultural collections, but a lot of those attributes that are at the record level are actually either private by default or can be made private by the collection. And those are things like value, monetary value, things like you would make sense you would want to keep, private to the collection. So we have a lot of those aspects, and we can entertain more specific requests as they come. We also have a mechanism to encumber entire records. So we have a lot of records that are just completely hidden from public view, and that's actually mostly for the NAGPRA purposes. So but collections obviously want to keep all of those things while they're being under reviewed. But again, if you're logged in, you can see all the data that you need to see. Any other questions? Yeah, go ahead. Sorry.
Also, we're I think it's in progress that we're also adding uses. So if you put, if you have very complex uses and permits, like that will show up, I think, in our search now,
that's
right?
already-
Yeah.
yeah.
So it's already implemented. So if you have very confusing, like it can only be used for this particular purpose, and everyone has to acknowledge specific people or national parks, etc., like that's enabled.
Yeah, that's a great question, though. Any other questions? Okay. All right, so these are for the users in the crowd. Or actually, you don't have to be a user at all. I just have a couple of questions I wanted to hear from you folks. And that would be, let's see, what are your favorite tools for working with tables or lists? Like Excel, OpenRefine, Legos, I don't know. I wanted to hear from you guys what sort of stuff that you're actually using beyond your collection management system. Or maybe you don't. You're like rely entirely on your collection management system, which I don't believe is true, but anybody? You like OpenRefine? Okay. Google Docs, yeah, Google spreadsheets. How many
For
Google
people on
spreadsheet
Zoom,
users
we have
here?
Google Spreadsheet, Excel.
Yeah, Excel. Okay. And OpenRefine, how many OpenRefine users do we have here?
We have some OpenRefine users.
Oh, good, okay. So if we were to build out specific services and apps and connectivity between Arctos and these external applications, which application would be most desirable or exciting to see? What? What did you say? Oh, Grubhub. Okay, yeah. DoorDash is not an option. I'm sorry. Yes, exactly. He wants to. Yeah. And the wrong answer is Excel macros, but anything else? Yeah?
I'm just thinking about, like, sometimes you want to visualize. It's nice to see, like, an agent page or a project page with all the images. You might want to visualize it as a graph.
Yeah. So.
Would that be easy to do, like export the images to some simple graphing software, like have like projects and
Yeah.
agents and specimens deposited?
Yeah, we are thinking about that right now. We're thinking. So let me ask you this: what kind of where would be the data point that you would want to see visualized? Like a search result or on a particular data page?
I think you can start with like the impact metrics, right?
Okay.
Like something you showed for like the PCP.
Mm-hmm.
How many specimens were deposited? How many publications and associated projects and loans were generated? Something just to show impact.
Yeah. Yeah, I've been thinking about.
It's hard to like download all that.
Yeah.
Excel or Google Sheets or whatever, and make a graph.
Yeah.
Of impact.
Yeah.
There must be some simple way to do it and show this.
Yeah, so we're starting to discuss and spec out a little charting widget, so to speak, a graphing widget, and so that's kind of the feedback we want to hear. So it sounds like you.
Like this thing.
Yeah,
There's some interactive like graphing software that produces this knowledge graph.
that's right.
Different nodes and show the number of specimens.
That's right, like a network. Yeah, yeah,
Because like
yeah,
getting to a point would be really hard, even with like really linked data.
Yeah.
towards markets,
yeah.
unless you really know how to make graphs.
Right, exactly. Yeah. So that's a great idea, but we hadn't talked about it from an agent impact, the activity report, but it wouldn't be difficult to do. So, yeah, that's a great idea. Anybody else want to jump in? Yes.
As far as like bulk loading records, it would be really nice to have some kind of feature where multiple people could be working on it at the same time. For those, we have multiple CAs working on the same bulk loading project. That's we had to do that over I think it was like some of us were using Excel. Eventually we got it together, but some of us were using Excel, some of us were using, messing in Arctos directly, some of us were using Google Sheets. So if there's one place in Arctos where we could be working on a bulk load together, that would be awesome. Does that make sense? Oh, I'm sorry. On like a say we were working on like bulk loading data rich records from our teaching collection or something. It would be nice for everybody to be, you mentioned like developing a tool directly like linked to Arctos to kind of mess with data or edit data related to your records. If that could be like multiple people working at the same time, that would be great. Well, no, multiple records at the same time.
Okay. Do you mean like in a collaborative way, like in Google Sheets?
Yeah.
Or like...
Yeah. Like basically Google Sheets, Google Sheets for like bulk loading a whole chunk of records, but being able to do it all at the same time. Right. So like your bulk load builder, but like we can build directly on that. Does that make sense? Okay.
I'm hearing Google Sheets, right? Okay. So yeah, well, just to piggyback on this, what other kind of data services would be useful to users besides data entry, bulk loading it collaboratively? What about cleaning your data? You saw some of the data quality tools we already have. Maybe if you're not familiar with all these things and you want to see more of these as a demo, you can come to one of the monthly meetings. I should say that Arctos working group meetings, even though we're supposed to be in summer mode and we're down to one a month during the regular season, academic season, we actually have two. We have one that's focused more on the issues and one that's more focused on the Arctos working group, sort of broader community. And we do demos all the time, and whenever we roll out some new things, we make sure we announce them and get great feedback from that. Yes. And separate meetings for each committee. And then we also have separate meetings for each committee. Yes. There's a meeting every day if you want to go. Or else my calendar would explode. But yeah, we can always show people more of those. But yeah, I'd like to hear from you guys about other. Yes. Hear from you guys more about the various services you'd like to see developed. Maybe something brand new. Maybe just something enhanced. Anything? Yeah.
This isn't data services, but it's one metric that I have to track outside of Arctos that I think we could track within, which is tracking visitors coming through the collection. It's something I have to tally every year. And so that could relate to agents if we know them, but many of them, it's just, a group of 14, and we might be classifying them by K through 12 groups or, fish and wildlife or something. But I always think about that because it's kind of the one thing I have to do by hand.
Yeah, I've actually been talking a little bit with the cultural collections committee to do something like that, because they have to track that for their NAGPRA requirements, their reporting. So yeah, basically just kind of like a log of visitors and maybe the specific things they saw. It doesn't necessarily mean, you could do it. Angie does do it right now, as you could do it as a loan, because it's. But maybe, just as tours, maybe we need another kind of transaction or interaction with the collection that's a little bit more general. Yeah, that's a good idea. So you should come to our cultural collections meeting, which usually interferes with my officers' meeting. Yes?
Yeah, so Arctos is an amazing collection management system, but the thing I worry about is when we direct people from our website to Arctos to look for samples or look for materials in our collection, I think it's really daunting to naive users. Is there some way to create like a collection-specific interfaces that are simplified and
Yeah.
just have the information most people are looking for?
Yeah. Okay. So this leads me to two things. One, to harken back to my super fast intro. So we are working on a couple of different kinds of search portals. One's based on Elasticsearch, and they will have one that's biologically focused, just the biological collections, and other cultural and geologic. And those will have filters, so you can filter by the indices that you like, or with the taxon search, if that's what's important. So those are in development, and I didn't get a chance to prepare anything for a demo of that, but if you're used to doing any kind of online shopping, you often see these filters, like filter by brand or whatever. So it'll function very similar to that. So we're developing the indices for the different collections. We don't have anything, like I said, specific to the cultural and geologic right now, but, we'll have those coming forward. And then this is my question to you guys, is to find out what is your discipline and what does your ideal search portal look like? Because we are looking specifically into those more user-friendly. I mean, they'll have links back to, of course, the actual records of interest, but these would be more focused on general searches, right? And that's the other focus of those assistive chatbot that we're trying out, which will take a natural language, process the natural language query, and then stick it in the right parameters. Our chatbot's still learning, still kind of figuring out what's a begin date and what's an end date and what's that sort of thing. But it knows country and counties really well now, and some taxon. We're trying to figure out if it can learn common names. So you could just say, how many raccoons do I have from Alameda County, for instance? And so they don't need to know all the specific search fields to stick that in. Yeah. Oh, yes, that's right. And if you truly need to have a very customized, standardized search portal, you can do what the Museum of Natural History at University of Alabama's done, which is team up with some students there, and they've developed customized search portals and specimen pages for based on the Arctos API. So that's actually available to anybody right now who wants to build out something specific to just their collection type. They can use the API. They just need to file an issue. We say this all the time. So yeah, just file an issue, and we'll take care of the API key and get in contact with you. So. But like I said, I realize that's also not necessarily an option for a lot of folks. So, like I said, we're building out some of these other alternatives, and those will just get linked on the homepage. So people, might still go to Arctos, but they can then go try one of the filtering searches, or maybe invoke the chatbot. Well, that's true. We do have saved searches, but that is, the curator could also run the searches. I do this all the time, and then I create a shareable URL and pass that link to the researcher, so they don't have to think about it. They just need to click on the link, and it'll pull up the same search results that I'm using.
Yeah, from the Zoom, Litsa Wooten, has asked if you've considered any of her improvements or suggestions for Berkeley Mapper, like live filtering without having to redo a search on the main page.
Yeah. Well, okay. I'm sorry. Who did that come from Zoom? Yeah. Okay, great. Thank you. I'm glad you're looking after our Zoom users. That's a good suggestion. I actually hadn't seen that suggestion right now. Right now, the way Berkeley Mapper works is it pulls on the temporary table that you've created from your search results. So within that, you can filter, you can, subset either spatially or through a few of the parameters that you searched on, all within Berkeley Mapper. But if you're going to do a brand new search, then that would require a brand new Berkeley Mapper instance. So we could figure that out, but it probably would have to be like a different widget than Berkeley Mapper at the moment. But that's a good suggestion. So stay within the mapping environment, I think is kind of what I'm hearing, and redoing searches, instead of redoing searches. Yeah. Okay.
Okay, I know I've repeated this to you 20 million times, Michelle, but most of my people that are requesting loans from me, they want to just be able to search on taxa, like a genus or a family, and tissues. They want to know, do we have a tissue for that thing? Or do we have like a DNA extract? Like that's the most basic thing they really want, to find it quickly. And it's a little, I think it is a little daunting for them to go to the Arctos search page now and have to do their own refined search, and they're like, I just want the button there. So that's my big request.
Carol, has tissues is the first thing we indexed on the new Elasticsearch portal. So yes, that is the one little toggle you'll have. But they'd have to go to the Elasticsearch portal, which is definitely a lot less daunting looking.
With what you're describing with that search portal, is it that you, like on our webpage, could we send users to that search portal? Or, okay, so it's not like they'd come to Arctos and then they'd have to decide which search portal to go to. You could direct that decision.
Yeah, they could go either. It's standalone. It
Got
just takes it's based on a snapshot of the filtered flat. So.
I have a suggestion.
Oh, yes.
I think yesterday Amy Briesman presented on local context, which is not a data repository, but allows tagging for Indigenous communities. And I think that would be really great for anthropological collections, but not exclusively.
Yeah, so we've been in contact with the local contacts for about, 5 or 6 years. We've both gone through our own organizational, growing up. So I think now's the time, now that we're kind of both over some organizational hurdles, to re-engage. And we've definitely talked about that because of the ethnographic collection at University of Alaska, because they are like the, cultural hub for Alaska. So yeah, we've talked about them. We've also talked about trying to include some native lands. They have an API right now for their geography. So yeah, we were looking at this from a lot of different angles, but again, kind of considering it so we can lower the burden of manual curation. But yeah, that's a great suggestion. And yes, I was very excited to see their talk yesterday too. Anything else? Do I have anything else on my list? Yeah, so any other portal requests or Arctos requests, and yeah, or search portal requests. Carol wants tissues. She always wants tissues though. Yes. Yes. Oh, are we at the break?
Yeah.
Oh, apologies, sorry.
Coffee time.
Never
Coffee
mind.
time. All right, well, thank you so much. You guys know where to contact us individually or as a group on GitHub, so we'll see you around. Thank you again. Oh, and yes, now you get your Arctos button. You survived the Arctos-verse, so don't forget your Arctos button.